Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump in, if you enjoy our deep dives, please do take a moment to like this video and subscribe. It really helps support the Sand Science Experience. Okay, so imagine a world where we could fix genetic diseases with pinpoint accuracy, or maybe engineer crops to survive tough conditions. Gene editing, especially with tools like CRISPR, promises exactly that. But, these tools, revolutionary as they are, often have drawbacks, things like unintended edits, unpredictable results. Today, we're diving deep into how artificial intelligence is stepping up, not just to use this tech, but to actually design it, making gene editing smarter, safer, and, well, way more effective.
That's right. We're going to unpack some really groundbreaking research published recently. Shows how AI was used to essentially invent completely new gene editors, highly functional ones. It's really pushing the boundaries of what we thought biology could do.
Okay, let's unpack that. We know CRISPR-Cas tools, the ones originally found in microbes, are incredibly powerful.
Oh, absolutely. Game-changing tech.
But when you take them out of their natural home, put them into, say, human cells, well, things can get complicated. They might not work as expected.
Exactly. There can be these functional trade-offs. Think of it like trying to use a perfectly good tool, but in the wrong environment. It might struggle, or worse, cause accidental changes, those off-target edits everyone worries about.
Right, the off-target problem. And I guess trying to manually tweak these natural tools has limits.
It really does. Traditional protein engineering, trying to optimize them, it often hits a wall. Biological systems are just so complex, and you're kind of stuck with the starting materials evolution gave you.
So this is where AI comes in differently.
Precisely. This new approach basically sidesteps those evolutionary limits. They trained large language models, kind of like the AI you might chat with online, but on a massive biological data set.
Interesting, like teaching AI the language of genes and proteins.
You got it. The AI learns the sort of hidden rules and grammar of how these CRISPR systems work directly from nature's own library.
So when you say massive data set, just how big are we talking here? What did they feed the AI?
Okay, get ready for this. They curated over 1 million CRISPR operons.
1 million?
And they mined these from an incredible 26 terabases of assembled genomes and metagenomes.
Terabases? That sounds huge.
It's astronomical.
Yeah.
A terabase is a trillion DNA bases. So imagine feeding the AI this gigantic atlas, they actually call it the CRISPR-Cas atlas, mapping out all known CRISPR systems from across the planet. It let the AI learn from just an unprecedented amount of natural diversity. See patterns humans just couldn't.
Okay, wow. But here's the really wild part. You mentioned the AI wasn't just analyzing existing stuff. It was, creating new proteins?
Exactly. That's the leap. These AI models were generating completely novel CRISPR proteins, designing them, essentially.
Like an AI biologist inventing tools.
Pretty much. And the results.
Yeah.
These AI-generated proteins expanded the known diversity of CRISPR-Cas families by almost 5 times. 4.8 times, to be exact.
Incredible. So the AI is designing things evolution hasn't even tried yet.
That's the key takeaway. It's exploring biological possibilities beyond natural evolution. But crucially, these new proteins still have all the important bits needed for gene editing to work.
And one of these AI designs really stood out, right? OpenCRISPR-1?
Yes, OpenCRISPR-1. They put it through rigorous testing, of course.
And how did it stack up, especially against the workhorse everyone uses, Cas9?
That was the big question. The results were pretty remarkable. OpenCRISPR-1 showed similar, sometimes even better, on-target activity. So it's good at cutting where you want it to cut.
Okay, so it works well, but the safety aspect, off targets.
That's where it really shone. It showed a 95% reduction in editing at those known problem spots, the off-target sites where Cas9 sometimes makes mistakes.
95%. Wow, that's not just an improvement. That's a massive leap for safety, isn't it? Especially if you're thinking about therapies in people.
Absolutely revolutionary. And what's fascinating is that OpenCRISPR-1 isn't just a slight tweak of Cas9. It's significantly different, like 400 mutations different. It's genuinely a novel AI-designed solution.
Amazing. So safer and truly new. Any other benefits?
Yeah, a couple of other key things for potential human use. It also showed lower immunogenicity compared to Cas9.
Meaning less likely for the patient's body to attack it.
Exactly. A lower chance of immune rejection, which is a big hurdle for gene therapies. Plus, it turned out to be compatible with another precise technique called base editing.
Oh, right, where you change single DNA letters, so it's versatile too.
Highly versatile. It really expands the toolbox for correcting different kinds of genetic errors.
So stepping back then, what's the big picture here? What does this really mean for science, for us?
I think it signals a real paradigm shift. AI isn't just analyzing biology anymore, it's actively designing novel biological solutions. It can overcome the limitations of natural evolution.
Creating tools specifically for our needs.
Precisely. Truly fit-for-purpose gene editors. This ability, well, it could unlock incredible precision for genetic therapies, for making better crops, for creating biotech tools we couldn't even imagine before. We're moving from just finding nature's solutions to actively creating potentially superior ones.
An incredible look at AI, not just understanding biology, but actually building it. That really wraps up our deep dive into this groundbreaking world of AI-designed genome editors. We hope you found those insights as mind-blowing as we did. It certainly gives you a lot to think about regarding AI's creative power in science. Thanks again for joining us. Remember to like and subscribe if you haven't already. It helps the Sand Science Experience keep bringing you these explorations. Join us next time for another deep dive into the sources that matter.